we're going to focus on the special senses. Now, the special senses are going to be differentiated from the general senses. The general senses focus on touch. Those are your tactile receptors, such as temperature, pressure, and pain receptors that we learned in the integumentary system. But the special senses focus on smell, which is olfaction. This is a chemoreceptor. Taste, which we learned is gustation, also a chemoreceptor. Then we have sight, which is a photoreceptor, a light receptor. And then we have hearing. And then the mechanoreceptors, which affect balance and equilibrium, will also be dealt with and discussed as we talk about the cochlea of the inner ear. Now let's talk about the eye. The eye is the organ of vision. The eye is the organ of vision, and as we said already, these are primarily going to be photoreceptors, allowing for, we have some accessory organs. We have the eyelids, which is the protective covering. We can close the eyelids to protect the eye from anything coming at it. There is something called the conjunctiva, which is an inner lining of the eye. This is like the red of the eye. The conjunctiva easily gets infected with bacteria. This is then called conjunctivitis. Then we have the lacrimal glands or lacrimal apparatus. The lacrimal glands will secrete tears, and tears, this liquid contains an enzyme that breaks down bacteria. Crying, secreting the tears actually act as a antiseptic, a protection against bacteria and foreign invaders, to break down something that should not be there. Then we have the breakdown of the eye. You're going to have to label the eye. We're going to focus on the cornea, which is the covering, the aqueous humor, humor referring to liquid. This is more of a thinner liquid between the cornea and the iris. The iris is actually the color part of the eye. This is a colored muscle that will get larger and smaller. And as it gets larger, the iris gets larger, the pupil or space will get smaller. And as the iris gets thinner, the pupil gets larger. So the iris is a colored muscle. Then you have the lens, and the lens is held together by the ciliary muscles. Then you have the vitreous humor, which is the liquid on the inside of the eye. That's the bulk of the eye. This is more of a gelatin liquid. It's not as clear, liquidy, or thin as the aqueous humor. It is actually thicker. Then you have the sclera, which is the white of the eye. And then you have, and that's on the outside, and then you have the retina. And the retina is the tissue on the inside of the eye. It's very colored. It looks kind of like a ink spot on, maybe an oil spot on the road. And basically it has rods and cones, which rods provide black and white or a gray color, and cones provide actual color, your red, green, blue colors. And then you have the optic nerve, which actually runs to the brain. So here we have the outer tunic, or tunic means layers. So you have the outer tunics, which is the cornea and sclera. We said the cornea is basically a more of a transparent covering of the outside of the eye. If you ever have to put contacts on, you're going to put the contact on and touch the cornea. So the cornea is right in the front. The sclera is the white part of the eye. Then we have the aqueous humor. As we said, that's more of a thin liquid. And this is between the cornea and the iris and lens. Then as we move into the middle layers, we have the choroid coat, which basically contains a lot of blood vessels. This is more vascular. This contains a high level of blood carrying nutrients to the eye. We have the ciliary body, which are little muscles that hold the lens in place and basically will pull on the lens. And we have a term called accommodation, which is the process by which the lens will change shape as it's going to focus on closer objects. So these ciliary muscles will pull and maybe relax, causing the lens to change shape to allow us to focus on an object. Then we said the iris is the colored ring around the space, which is the pupil, and that iris is going to be a specific muscle that allows light in, or if it's too bright, will get larger to decrease the size of the pupil to let less light come in. So here we have the pupil, we have the iris, which is the muscle, and then we have the white of the eye, which is the sclera. Then as we move further into the eye, we have the vitreous humor, which is the second, we said thicker liquid, really makes a bulky shape of the eye, allows the eye to create its round shape so it doesn't collapse inside itself, and it allows for light to pass through. Then we have the inside layer, which is the retina. And the retina, as we said, contains rods and cones. Rods, which are your gray tones. And then we have the cones, which allow you to see color. We're going to take a look at an eye chart, a Snellen chart, when we do our labs in class this week. Here we have astigmatism. Astigmatisms are going to be an unevenness of the cornea or the lens, which causes a blurriness or blurred vision. Now we're going to move into the sense of hearing. We have the external ear, which what we usually pierce the ear with, that cartilage structure with the earlobe that's kind of like a sonar dish, that is called the auricle. Then the ear canal, as we go into the ear, is called the external auditory meatus. And this ear canal moves through the temporal bone. And it does function to start the vibrations of sound waves, and that allows it to be transmitted towards and interact with the eardrum. Now, the eardrum is called the tympanic membrane. You might have remembered this with biology as we dissected the frog, the bullfrog. So the eardrum is the tympanic membrane. Now the tympanic cavity is a cavity right behind the eardrum, and this is actually filled with air. Then connected to the eardrum, connected to the tympanic membrane, we have the auditory ossicles, which, remember, ossicle means bone. So we have a total of 3 auditory ossicles, 3 middle ear bones, which are the malleus, incus, and stapes. So the malleus connects to the tympanic membrane, then the incus connects the malleus to the stapes, and then the stapes connects the incus to the oval window. And basically the oval window is the entrance to the inner ear. Now we're going to be talking about the inner ear in just a second, but then we have the eustachian tube or the auditory tube, and this is a pathway that connects the middle ear to the nasopharynx. This is what is actually you're going to feel when you go on an airplane. You feel like you have bubbles in your ear and you have to kind of hold your breath or swallow, and you hear the popping. So if you have maybe a cold, the eustachian tube will kind of swell up, causing it maybe some crackling or some pain in the ear. So the eustachian tube basically equalizes pressure. So when you go up in the airplane, there's going to be a pressure change. So these air bubbles are going to kind of move through. So your eustachian tube definitely is needed in our bodies to relieve pressure. So here you can see the 3 middle ear bones and how small they are. You have the malleus, incus, and the stapes. So the stapes kind of like a structure that when you go on a horse, you put your feet through on the saddle. Now we're going to start moving into the inner ear. And as you can see, we talked about the outer ear, the middle ear, and now we're moving into the inner ear from the oval window. And this is actually where we are going to find the organ for hearing. And the organ for hearing is called the organ of Corti, and that's in the inner ear. So there's going to be some different labyrinths or complex systems of intercommuning chambers or tubes and those are called labyrinths. There's the bony labyrinth or osseous labyrinth and the membranous labyrinth, which basically the membranous labyrinth is like the osseous labyrinth but with a membrane. Okay, so that's pretty easy. And you have two different types of liquids, and the liquids referring to lymph is the perilymph, which fills the spaces between the osseous labyrinth and the membranous labyrinth. Now, the endophil fills the membranous labyrinth. So the perilymph fills both, endolymph fills the membranous labyrinth. Now, this is the important part of the inner ear that I need you to be aware of. You have the cochlea, the semicircular canals, and the vestibule. So the cochlea is the snail-shaped structure. The semicircular canals are the three rings. The vestibule is just what's between them. So the vestibule is the area between the cochlea and the semicircular canal. And this allows for static equilibrium. So let's take a look at those three parts. So you can see the semicircular canal, you can see the vestibule in the middle, then you see the snail-like shape, and that is the cochlea. So here's the cochlea, the snail-like structure. Now there's two compartments of the cochlea, and they're both filled with the perilymph, and that's going to be the scala vestibuli and the scala tympani. And basically these are just compartments, an upper compartment and a lower compartment. So we said the organ of Corti is the mechanoreceptor, the actual organ of hearing, located in the cochlea in that snail-shaped structure. Now you also have, you can see right here, you can see the cochlear duct, you can see the different chambers here, you can see the perilymph, the liquid inside, and then you have this basilar membrane. Remember we talked about basal membranes with the integumentary system. We talked about the different tissues or cells and the layers. Well, basically what you're going to see here is that when we move our body, you have this membrane that does move, and you have these little kind of crystal-like rock structures on the very top called otoliths, and you have little hair cells. And as we move our body and we move our head, these otoliths will move in different directions. This gelatinous material will move because of gravity, and that allows us to really be able to identify, are we being tipped over? Are we not straight up? This gives us a sense of equilibrium and a sense that we are not. So this is the basic pathway. You will need to know this. It's pretty easy if you go over it and just repeat it a couple times, practice it. So we're going to start with the pathway of sound waves entering the air. So we have number one, the auricle, which is the cartilage sonar dish. Then you have the external auditory meatus, which is the air canal. Then you have the eardrum, the tympanic membrane. Then we go malleus incus stapes. Then we go oval window. Then we go perilymph of the scala vestibuli, endolymph. Then we go hair cells in the organ of Corti. Then we go cochlear branch, which is a nerve from the air. So now we're moving away from the air, and we're moving into the brain, and we move into the thalamus, and then cerebrum. So this is how we are able to identify the stimuli, so the input to the central nervous system. Okay, now we're going to move into the sense of smell. We have olfaction. Olfaction or olfactory receptors are at the roof or top of the nasal cavity. Now the focus here is these are chemoreceptors. These chemoreceptors, these chemicals, need to be dissolved in moisture, and basically we're able to identify through these neurons with long cilia basically the different smells. So the body is able to interpret the smells, and this is then transmitted these neurons are transmitting the stimulus to the temporal lobe or the olfactory area. So the olfactory area is in the temporal lobe. So you can see at the top of the nasal cavity, you have these chemoreceptors, and basically these chemoreceptors are like these long little neurons with little hair-like structures on them. And basically they send the impulse, okay, to other neurons that will then eventually send it to the central nervous system. So these odor molecules do need to dissolve in order to be picked up by the olfactory cells. Okay, lastly, we're going to talk about the sense of taste. You have taste buds. Basically, these are clusters of nerve endings on the tongue. Now, taste buds don't just reside on the tongue. Most of them are on the tongue, but they're on the roof of the mouth. They're in the cheeks. Now, specifically the tongue, we do have something called papillae, which are little projections. They are lifted up. There's some that have taste buds on them and some that don't. So the filiform papillae don't have taste buds, but the fungiform papillae are rounded with taste buds. And so basically these papillae may be different shapes, different sizes, and basically they will have little receptors inside them that actually send. So here you can see the fungiform, the filiform. So the gustatory cells are basically the taste receptors. These little long hair-like structures or microvilli, they're like little fingers that absorb different materials. Specifically, they're going to be stimulated by chemicals. So these are chemoreceptors, and they have to be dissolved in saliva or a liquid. And basically, this then sends the impulse to the parietal lobe. So basically, you can see the taste bud structure with the papillae on top. You can see the little gustatory hairs or microvilli. Some resources say that there are different taste regions, like there's a sweet region, a salty region, a bitter region. Now, some resources say no. Some resources say it's just what your brain is interpreting based off of the absorption of the chemical at the microvilli. So whatever, it's taken wherever, and it's just being translated that you're tasting it at that certain region. Now, I am going to share with you the map of the tongue, and you will need to know that, even though there's some debate about that. So you can see the basic structure of the tongue. You can see where the different papillae would be located. And you can see here's a basic structure and outline of some of the taste sensations. So with sweet receptors, you got sugars, primarily sour, dealing with acidity, bitter, going on the opposite of that, your alkaloids, and then salty, which would be your metal. Now, kind of in the middle there where we don't have anything, that could be all other tastes. It is described as something called umami, and this is kind of not really salty, but this is a different type of salt, more of a compound that's going to cover all other tastes. So we have the sweet on the tip, the sour on the sides, salty primarily covering the perimeter of the tongue, front tongue. Bitter is going to be the back of the tongue. And the umami is pretty much the center covering everything else. So identifying all other taste.